Hi everyone, in this module we'll be extracting the DNA from a strawberry. And we'll also be learning some basic chemical procedures that are regularly used by chemists and bioengineers. In the previous module, we made a model of the shape of DNA using pipe cleaners to see the double helix shape, and in this module we'll be able to touch and see real DNA. We can use strawberries for this experiment because all living things, including strawberries, which come from plants, contain DNA. If you don't have strawberries, that's totally cool. You can also use human cheek cells. We will also be letting you know how to use human cheek cells and how to get them safely for this experiment. Also, this experiment is super fun to try at home and I really encourage you to try it. There are definitely a lot of steps, so don't feel discouraged if you don't get it the first time. Even in the real world, scientists have to work out their procedures through trial and error. I'm sure they failed many times before getting it right. So for this experiment, you'll need strawberries, or you can replace it with human cheek cells, which don't need any material except for yourself, one small resealable Ziploc bag, dish detergent, salt, water, a coffee stirrer, or any other type of stick, a coffee filter, a disposable cup, rubbing alcohol, and newspapers or something to lay across the surface to keep it clean. Our first step is the physical breakdown. We will be physically breaking down the strawberries using our hands. First, place 2 strawberries after removing the green leaves into the Ziploc bag. Push out any air and seal. Mash the strawberries through the bag using your hands. You can also pound them with your fists if you want to. If you don't have strawberries, we can use cheek cells instead. Instead of putting strawberries in the plastic bag, pour 10 milliliters of water into a cup. Swirl the water around in your mouth for 30 seconds to a minute, and then spit the water into the plastic bag and seal the bag. The second step is to make the lysis solution. Lysis means to break open. This step is important because DNA is within the cells of an organism and we must break open these cells in order to release the DNA into the solution and isolate it away from everything else. To make the lysis solution, you need 2 teaspoons of dish detergent, 1 teaspoon of salt, and a half cup of water. Mix these together, but try to avoid making any bubbles. As you can see, I did make some bubbles, so don't worry if you make some, but try to avoid making bubbles as much as you can. Add 2 teaspoons of the Lysis solution, or maybe a few more if your spoon is small, into the plastic bag with the strawberries or with the cheek cells. You might hear some popping sounds. This is because the cells are being broken open, kind of like popping a bunch of tiny water balloons. The Lysis solution is able to break open cells because the dishwashing soap is able to dissolve the cell membrane that surrounds the DNA, and the salt in the Lysis solution separates the proteins from the DNA. The third step is the filtration step. If you are using human cheek cells, you do not need to do this step. In this step, we'll be using the coffee filter in our empty cup. Place the coffee filter into an empty cup. Open the Ziploc bag and pour the contents into the cup through the coffee filter. Pick up the coffee filter, twist, and gently squeeze to ensure as much of the juice falls into the cup as possible. The pulp in the coffee filter can be discarded as the DNA is in the filtered liquid inside the cup. As you can see, here I have the liquid that's been separated from the pulp in this cup, and that's what this filtration step is for. Our last step in this experiment is the precipitation step. You will need rubbing alcohol for this step, and remember to be really careful with rubbing alcohol. Pour the rubbing alcohol down the side of the cup such that there is a layer of rubbing alcohol above the filtered strawberry solution. Please remember, do not stir. You will observe a cloud of white strands gradually appearing in the upper alcohol layer. It might be a little hard to see in this video, but if you try it out for yourself, it might be more clear without the glare and without the bad video quality. Use the coffee stirrer to remove this DNA. As you can see, this DNA that's extracted might not look like the model you made in the previous module, but this is just to let you know that in real world, the DNA looks like tiny thin ropes, just like shown in this module. Also, as some background information for this step is called the precipitation step because just like how rain precipitates and falls from clouds, the alcohol makes the DNA fall out of the solution and appear as a solid state so that we can observe it. Also, remember at the end of your experiment, save any unused reagents and remember to dispose of any used reagents. Be sure to clean up, and I hope you really enjoyed extracting DNA out of a strawberry. And that's it for this lesson. Thanks for participating in BEAM, everyone, as this is the last lesson of this semester, and I hope everyone had a lot of fun. Thanks, and congratulations on this semester of BEAM.